Okay, students, since we don't have any physical class today, I'm just gonna do a class recorded for you. So, an important part of biology is to be able to draw and recognize structures of cells and realize what the different parts of cells do. So this is something we're gonna spend some time on. And today in total we're going to look at 3 different types of cells. So in that way we're going to learn how to differentiate them, and that will help us also to understand a bit of their different functions and ecologies. And the first one we're going to draw together today is the animal cell, and then on our separate lessons on Monday we're gonna draw the plant and bacterial cell. So let's see how this goes with this software. So the first thing when we draw a cell is, of course, drawing the outline of the cell. So I'm gonna do my best to make my... Ooh, look at that. That is the outer part of a cell, and that would be the cell membrane. Ooh, where did that go? I wanted to move you. I guess I'll do that again. So this is the cell membrane. Sometimes you call this also the plasma membrane. Let's see if I can move this thing. I can. We can also make a little line to show what we mean. So cell membrane or plasma membrane. So the plasma membrane is what we're going to write on the side here the functions of it. So the cell membrane is composed of a special type of molecule called phospholipids. I'm going to say mainly of phospholipids. We're going to learn way more about these later, but the cell membrane separates the internal and external environments. Of the cell, and it's really important for the transport of substances from inside the cell and out of the cell and vice versa. So main functions include transport, but it also has the ability with special proteins in the membrane to sense the external environments. So for example, your cells that receive hormones from other cells, for example, your adrenal glands close to the kidneys will secrete hormones into the bloodstream, and they will attach to the proteins on the outside of or in the cell membranes of other cells, and that will change how those cells function. Communication then is very important, and sensation and reaction. So you also have in the plasma membrane, you could have receptor proteins that respond to certain chemicals in the outside environment. Your light sensitive cells in the retina, they have special proteins in the cell membrane to give that ability to sense light. By doing this, we also find, of course, or we also more or less show the inner part of the cell, and that's called the cytosol or cytoplasm. And that's the liquid part inside of the cell here. I hope you see this arrow moving around. So that's the internal environment here. So the cytoplasm is mostly water. And as you see, I will use the different words completely interchangeably. So sorry about that. But a cytosol is the liquid part of the cell. And it has mostly water, or it is mostly water, and dissolved substances. so ions, salts, and so on, but it also has proteins, sugars, and many things like that. And enzymes, let's say. So enzymes are specific proteins that do chemical or that catalyze chemical reactions. So they're really important for the metabolism of the cell. So the cytosol is important for transport within the cell and for metabolism. So chemical reactions such as respiration happens here. At least the anaerobic part. And respiration, we're gonna learn way more about that in the later part of the course here. But just write it down now, and you can have it for your notes later. Now, since this is an animal cell, we forgot to write that, so we're going to write that as well. And just for everyone, please continue. Anime. Oh my god. Animal cell. I do get emails from them, so maybe that's why I wrote that. But animal cell model, that's what this is. And of course, you should be drawing this as well. So practice for yourself. There we go. Okay. And since this is an animal cell, that means that it's also what we call a eukaryotic cell. Eukaryotic cells are special, or in that they have, all of them, a nucleus inside. So we're going to draw the nucleus as well. That's going to be the next part. That's this one here. No, that wasn't as beautiful. Let's do it again. I want to make a complete, perfect. Oh yeah, that's much better. Okay, so inside we have the nucleus here, and usually we can tell or can find a specific part of the nucleus like this as well. I'm just going to draw that too. And then I'm going to make it a little bit, I'm going to try to make a filling in here, but I don't know, maybe there's a way to do that that's much better, but I don't know. Like that. So the stuff inside is usually when, as you remember from your own microscopy of your own cheek cells, you saw the nucleus is something very heavily stained, of course, so that's why I draw it like this. And the thing here is called a nucleolus, and that's something you often see here. But nucleus, and a nucleus is always found in eukaryotic cells, like animal cells, plant cells, fungi, and so on. So eukaryotic cells are the ones with a nucleus. That's what it more or less means. Eukaryon is Greek for nucleus. So the nucleus is surrounded by its own membrane. And it contains the DNA of the cell, found in all eukaryotic cells. And inside the nucleus we have important reactions. Are replication and transcription. So this is, let's say replication is for copying DNA. And transcription is first part of producing proteins. So these ones we're going to learn way more about later as well. But so the nucleus, the main job of the nucleus is to make a special molecule called mRNA that outside in the cytosol is important for creating proteins. But it's also has a function inside that's called replication, or a chemical reaction inside called replication. That's making double the amount of DNA, and that's very important for cell division, so that both cells that are created from cell division have the right amount of DNA. So make copies first and then split, then each new cell has one part of the copy, or one copy each. And then around, let's see what we can make here. Why don't we make your favorite little thing? It's the mitochondrion. So the mitochondrion is kind of a special piece of the cell in that it has two membranes. It has one inner that is heavily folded like that, and an outer one going around it. Like that. And of course you have many of these inside one cell. So we're going to draw two to symbolize many. Remember that these are only models of cells, not actual cells. But you can recognize them always by this funny folded inner membrane. So these are... Wait. Now it disappears. Mitochondria. Mitochondria is the plural, mitochondrion is the singular of that word. Let's see, I keep doing that. Ooh, I'm not making straight lines. IB will be very sad. I wish there was a ruler in here. I know there was a ruler there, but I think it's not for making straight lines. Anyways, so the mitochondria. They have double membranes. Two membranes. One outer and one inner that is heavily folded. And the mitochondria are of course involved in respiration. And they do the aerobic part. So respiration is about generating ATP. ATP is the unit of energy that cells use. Whenever you say that the body uses energy to do something, it breaks down ATP. And we generate ATP with the cytosol and the mitochondria with respiration. And by breaking down fats, sugars, and so on. Let's see. Mitochondria are cool in that they have their own DNA. And ribosomes. And these are 70S. And you're like, what is he talking about? 70S? That's a specific type of ribosome. So we bring that in here as well. So then let's, this feature that they have their own DNA and ribosomes comes from the fact that mitochondria are most likely bacteria that have entered eukaryotic cells. So in the evolutionary history of eukaryotic cells, an important event was when bacteria were entered these cells and were engulfed by the outer membrane, which is probably, this is why I drew this one black, that represents the outer membrane of the animal cell, but the inner membrane has more bacterial-like structures. And the idea is that they weren't broken down and killed, but instead generated ATP and for some reason were not killed, were resistant to being killed within these animal cells. And then they started to cooperate with each other. So these ones were able to have a safe environment inside animal cells and got sugars and so on from the animal cell, and then generated ATP for the animal cell. So it's like win-win for everyone. And that's a symbiotic mutualistic relationship, but that's something we'll get to later. Okay, so next, and of course, please pause and take your own little breaks whenever you want during this recording. But around the nucleus, we have also a very interesting system of flattened sacs, or they look more like discs like that. Very difficult to draw, much smaller than this actually, but I'm trying as best as I can to make them beautiful. I'm so concentrated. But flattened discs like this, so they need to make, yeah, that's fine. This is my second favorite piece of animal cells or eukaryotic cells. This is the endo for inside, plasmatic. So it's inside the endoplasm. Plasmatic reticulum. So that means that it's like a network. That's sciencey cool word for network. So that's this part here, endoplasmatic reticulum. Endoplasmatic reticulum. And this is a membrane membrane sac system. Surrounding the cell. And there is a smooth endoplasmatic reticulum. Creates lipids for e.g. cell membrane. The rough endoplasmatic is bound or is coated with ribosomes and is involved in protein synthesis. Blah, blah, blah. And that's that. Okay, so we're gonna draw these little, make the rough part by putting little dots on the outside here. These are ribosomes coating the outer part of the endoplasmatic reticulum. This is better with this software than it is on my, when I do it on the blackboard, or whiteboard, sorry. But yeah, so they're coated, these small units here, they are ribosomes, but we find ribosomes in other parts of the cell as well, in the cytoplasm. So let's draw a few more. Whoa, that one was ugly. Maybe I should just make, look at that, what a clever cat. So those can be ribosomes then. So ribosomes, as you see, are very small. Let's see, we take the small pen again. Bam, and we take like that. No, that's not what I wanted to do. Right down here. So ribosomes are small structures, and they're made by two different pieces, two subunits. And I'm actually gonna write that these are 80S ribosomes. Made by two units, the small one and the big one. They're called the subunits. And they are involved in protein synthesis. Some in ER. That's the, we're gonna see if we can we write this? That one's called the ER for short. Some in ER for that's proteins for cell membrane and outside of cell. So cells generate proteins that are either bound in the membrane or is excreted out of the or secreted out of the cell, and some are instead in the cytosol for generating proteins and enzymes inside the cytosol. So others in cytosol for proteins in the cytosol. There we go. What else do we have? Then we're gonna draw something else, and they're actually way bigger than, or sorry, they're like all over the cell, but we're not gonna draw them all over the place. Let's see, what's the next one? Ooh, now here comes my favorite part. So let's draw this over here, actually. This is the Golgi Apparatus, it's called. I don't know why I'm doing this in green, but I just wanted to make it look a little bit different. So that's also another membrane sac system, but it's usually kind of made of these more folded sacs, or sorry, bent sacs like this. I'm trying to make them slightly more bent at least. And it's usually a system that's far away from the nucleus. So it's similar to the endoplasmatic reticulum, but it's further away. That's basically... So this is called the Golgi Apparatus. And that's from a famous scientist, cell biologist. He's also known for making his own stain for... So you will be able to look at this afterwards for yourselves as well. I'll give you the link to this. So the Golgi apparatus is a membrane bound or membrane sac. We did this before, so let's do it again. System far away from the cell. Not the cell, nucleus. So this system, so let's say we're gonna look back over here before we say something. So imagine this one is involved in protein synthesis as well. So basically you have ribosome here generating, synthesizing a protein, and the protein ends up inside, so it's generating it inside of this membrane system, the endoplasmatic reticulum. Eventually, a piece of this will bud off and move throughout the cell as a small little ball with protein inside. I'm gonna make this blue. So we're gonna make a little bud like that, and so these are what's called small vesicles. Inside them we can have protein. Let's make protein as a small little thing here. Beep, beep, beep. And it ends up in here, and then it is transported into the Golgi. And here the Golgi apparatus kind of this fuses with the membrane sacs here, and then they are kind of matured here before they are sent to the membrane. So basically you make several parts of a protein with different ribosomes here. They all get sent here, and then they get assembled and matured before they get actually sent to the plasma membrane. So all these membranes here are the same kind of structure. That's why they can be fused with each other. I know this is slightly complicated, but that's how it goes. We don't have to learn all the different things. I'm just saying this so you can have something to look back to later. But what we need to know is be able to determine what's the Golgi apparatus in the cell, and that's this membrane system all the way over here. So it accepts vesicles with protein from ER, matures proteins before sending them to the final destination, the cell membrane. So as you can see, these two have some kind of postal service or transport. Wow, my God! So this is our factory over here. The ribosome sends it to a local. Let's say that it's I'm the cell membrane. I've ordered a protein from I don't know. Let's say Germany. So this is in Germany. This is someone on Etsy making a bracelet that I'm really interested in for some reason. And they build it and they send it to the local DHL office. The local DHL office sends it over to Sweden. Here we have PostNord. And they take the package, that they put another label on it, and then they ship it away to the final destination, me. So that's kind of how we can see it. Okay, what else are we missing? Oh yes. Whoops. We have, inside the cell, we also have a very interesting little thing. We can draw them as this, long lines like this, and a second one that looks like that. So this one is a unit of, so these long lines here are protein lines, and these ones is the same thing but looking from another angle. So there are two of these usually. So each of these are the same thing, but imagine they're like barrels of protein. Imagine penne pasta or something like that. This one is on the side, this one has a front view down through the tube's little hole. And those are called centrioles. And oh, look at that line. So the centriole is very important for cell division. So when the cell is about to divide, the DNA kind of condenses into visible chromosomes, and we send protein strands from this one to this copies itself. So we have two of these. One goes over here, one stays over here. Together they are called the centrosome, and so they move to opposite side of the cell, and then they send different protein strands to the chromosomes in here and separate them in mitosis. They also help separate the different parts of the different organelles in the cell. So we have two different ones. So very important for cell division. We go down here and we write centriole. Oh, the noises you hear are my cats. Centrioles are protein units important during cell division for separating the chromosomes, that's DNA, for each daughter cell, also for separating or dividing the organelles between the two daughter cells. And yeah, What else we got? What else we got? So you can always go down and look for yourself here. Oh yeah, one more thing here that we're gonna draw. So sometimes we also have, or we tend to draw, we have other things here. We have, this is a small membrane sac that we call the lysosome. So lysosomes are small special membrane sacs in the cell, specifically in animal cells, and they contain dangerous proteins that are good for digesting parts of anything. They're good for breaking stuff down. So, for example, if this cell is a bad cell, maybe it's been affected by a virus and needs to be destroyed, the lysosome will actually break down, and all these digestive enzymes go out into the cell and start kill the cell from inside. So it's called apoptosis. The cell dies by itself. Or they can be used for when the cell engulfs a bacteria. So for example, a bacteria would be a smaller piece, would be usually smaller, and it can be taken into the cell by having the membrane here kind of wrap around it and bud off inside. And this one can fuse then with this budded off cell membrane big part and break down the bacteria inside, and then the cell can extract the different parts, so basically eating a bacteria. It can also fuse with a mitochondrion if the mitochondrion is bad, and then the mitochondrion will break down. So it's like a housekeeping or something like that within the cell. So, and as some authors say that lysosomes are unique for animal cell. So lysosomes, let's bring this to its own part here. But membrane sacs with digestive enzymes. So they can be used to break down broken organelles, digest phagocytized bacteria. Phagocytosis is what it's called when a cell kind of eats something from outside and envelops it in its own membrane. The animal cell will envelop the bacteria in the animal cell membrane, or to kill the cell itself. Apoptosis. Once again, these things you'll learn more about later, but that's kind of what it does. And usually you can tell a lysosome that there are other vesicles inside, so how can you determine which one is which? Usually the lysosome is bigger and it's stained more heavily, like that it will attract stain and looks darker usually. But I think we will end here with this lesson, because it's going to take you way longer than it took for me to write all these things down. But please draw this for yourself, and we'll continue with the animal and with the plant and bacterial cells, and you can always ask questions about these ones when we meet each other on Monday, and we will just continue with, and if there's something we need to add, we'll add that on Monday as well. But then the other two cells, we won't have to write all these things down necessarily, only the new things. But please go in here and find, if you haven't already taken all the notes, you can come back to this and do it whenever you want to. So I hope you will have a nice weekend. I look forward to seeing you on Monday. See ya.